Thank you very much for that introduction, and thank you very much, Virginie and Abdu, for inviting me. It's a great pleasure to be here, and it's a good place for me to give my talk because Abdu just gave a very nice introduction to sexual conflict and its importance for evolution of traits in the sexes and how sexes adapt to one another. So I'll pick up from there, and my focus in this talk is to summarize what we've learned about sexual conflicts that involve that are involved in life history evolution. So we're talking now about complex polygenic traits. And before I get there, I want to actually pick up on a little bit on what you just were discussing and talk about these two different flavors in which sexual conflict plays out. Let me see if I can. Yes. So Abdu talked about these coercive traits in males that harm females, and this arms race this sexual conflict between traits expressed by two different individuals, males and females. How that leads to arms race between the sexes. And so in the seed beetle system that we work on, here's a CT scan image, where is the pointer, sorry, of it. The extreme sexual conflict trait is this male adigas here, and it doesn't take much to see why that causes fitness reduction in mating in females. But the other form of sexual conflict that my work mostly touches on is this intralocus sexual conflict. So this is a conflict when a homologous trait is expressed in males and females and selected in antagonistic way. So shared alleles that males and females carry are selected either for or against in each sex. So this how this conflict is resolved is then what leads to the evolution of sexual dimorphism in a given trait. So we've got this sexual conflict that affects different traits in males and females, but then in order for these traits to evolve sexual dimorphism, you have to also resolve this other conflict which operates then within genes between alleles differently favored. So here we've got two different fitness peaks for males and females. And in order for them to achieve these, you have to somehow resolve this. Here we've got two alleles that are differently favored in the sexes, and how we sort of get the males and females to reach these different optima, given this conflict over the shared alleles is the question. And so what I want to do now is to first give a broader context to life history evolution in seed beetles and how male-female coevolution plays into this, and then take you to within populations to show you how sexual size dimorphism, so in body size, can evolve, because sexual dimorphism really evolves as a within population process. Then we talk about our experimental work to show how that can come about. And if I have time, I'm going to show you a little bit about the sort of genomic consequences of sexual conflict as sexual dimorphism evolves. Thank you. So life history traits don't evolve in isolation, they evolve in concert with one another, and this is because of the physiological constraints and trade-offs between different traits that have to get resources put into them. And so what we've recently looked at is how life history evolution plays out in the context of seed beetle phylogeny. We've looked at 12 different species and a bunch of different life history traits and how they co-evolve in males and females across the phylogeny in interaction with traits that affect the economics of reproduction. So cost and benefits of these cost and benefits of mating and in the context of this sexual conflict. And also how the sort of major sort of determinants of the ecological aspects play into this. So we've got here 11 different life history traits, 17 reproductive traits, 12 species. And just to sort of show you briefly the ecological side, our seed beetles, they utilize legume seeds as hosts for the larvae, and they can either utilize tree seeds or herb seeds. And what we can see is that this dichotomy has a big effect on their life history evolution. The species that have adapted to trees, they show this slower pace of life history, and this is true for both sexes. And so we've got a coevolution of metabolic rate, lifespan, and body size, and these host species kind of determine the availability of resources, and which determines population densities, which turns into effects on favoring either faster or slower pace of life syndromes. But then we also see this coevolution with different mating traits, and what we've observed is that species that have a larger body size and longer lifespan, they have slow metabolic rate, and they show a higher mating cost of males and lower mating cost of females. And this is really interesting because it suggests that there actually is no really a real resolution to sexual conflict when it comes to these mating interactions, so that as male cost of mating increases, female cost of mating decreases, and vice versa. And what is really important in this system is ejaculate volume, because this is a resource for females in these tree species. Females actually get nutrients from these ejaculates, and they put this into their reproduction, and this has turned the whole mating system to be dominated by female sexual selection. Whereas in these species with the faster life histories, it is the males that are more sexually more that are subject to strong sexual selection, and females suffer more from the cost of mating. So the life history, so the body size evolves in concert with this pace of life. And we see these similarities in the sexes across the phylogeny, but we also observe that the evolutionary rate for these life history traits is faster for males than females. And this brings me to this other conflict within genes between alleles, because we have to explain somehow how is it that males and females can show different evolutionary rates for these shared genes. And so we see it in the metabolic rate, which evolves faster in the males, and we also see it in male body size, and that actually is a player here in determining variation in sexual size dimorphism across this phylogeny. So even closely related species can differ, being strongly female biased in size dimorphism, and then losing the sex bias, sexual dimorphism, because of variation in male body size. So males and females share most of their genome. They are, in this system, they are XY species. This shows the karyotypes, and polygenic life history traits are, of course, traits where we expect to see this interlocus sexual conflict being more prominent, because the males and females should share most of the genetic architecture for such traits. And in fact, when people have tried to artificially select on body size in order to try to create some sex differences, it has turned out to be quite a hard thing. So these studies show here, these are in Drosophila, they show that if you select on only one sex, the other sex follows. So there's this correlated body size evolution in the other sex, and no dimorphism evolves. Then a recent paper showed that you can get dimorphism to evolve if you select sexually and ethnically, but it takes a really, really long time, suggesting that segregating genetic variants has often very similar effects on males and females, and it's really hard actually to evolve dimorphism. But at the same time, if we take a look at the animals around, we can see that size dimorphism is really, really common. So obviously there are resolutions to this conflict, but it's a dilemma how these kind of microevolutionary changes can happen, given that the architecture is so similar in the sexes. So what we wanted to do is to take our shot at the artificial selection and see how evolvable is sexual size dimorphism when we apply sexually antagonistic selection or just selection on one sex. So the same way as you saw in those examples. But with a twist here, we applied our selection on families, and what is really important here is for the sexually antagonistic selection is that we selected for families that show largest size difference between sons and daughters as a way to try to screen the genetic variants that allow as large difference in the expression of the shared genes between the sexes as possible. And so we did this for 10 generations, and I'll show you in a minute the results. And then we followed this up with looking at the quantitative genetic architecture of body size. So we looked at the genetic variances before and after selection, making a 3-generation pedigrees where we can partition additive genetic variances from dominance variances, and we can look at sex-linked genetic variances separately from autosomal variants, to understand how similar the genetic variance effects are in the sexes. And I will talk, if I have time, more about the follow-up studies. So we managed to generate very different sized beetles with this approach, and I forgot to say this species is one from the phylogeny, it's Calosebruchus maculatus. But we did not manage to generate sexual dimorphism in whatever way we applied the sex-specific selection. So if we selected only on females, no dimorphism evolved, as you see here. But body size did evolve. It's just that the males showed a very strong correlated response to the selection on females only. But if we selected on males, we saw that dimorphism either decreased or increased depending on the direction of selection. So we see that here. So we got about 30% increase or decrease by just selecting on males only. Then, here are the actual body size responses and how we see that the body size dimorphism actually evolved is because there was this time delay in when females responded to this selection on males. So this correlational response kind of kicked in after a few first generations. So this already suggests that there is something that can rapidly change dimorphism that is only accessible to selection when selection operates only on males, but that it can also rapidly fix. And you'll find out soon what it is. When we selected sexually antagonistically, we got a whopping 50% increase in size dimorphism. So really, we got a really rapid response, and this was because we actually saw a change in body size only in males, whereas the female size did not change. So clearly, this assay selection managed to kind of constrain the female correlational response, but also there is a constraint in dimorphism here too, because the female size did not manage to go upwards. So if we then turn to our quantitative genetic analysis, we see that overall males and females share the effects of the genetic variance shows a very strong correlation between the sexes, 0.92, the intersex genetic correlation here, although there are overall different amounts of additive genetic variance between the sexes. But this overall high genetic correlation between the sexes explains the correlational responses. But what explains the rapid response when selection could operate on males is that we found also strong Y-linked genetic variance in the males. And this is really surprising because the Y chromosome is really small in this system. It's less than 2% of the genome. We don't expect this kind of highly diverged Y chromosomes to harbor additive genetic variance. So this was a big surprise to us. And to really nail it down, we took males from each of these selection lines and we we crossed them with females from a single inbred line that was generated from the ancestral population. And we repeatedly backcrossed males to that inbred line in order to basically isolate the effect of the Y chromosome from the rest of the chromosome. So we replaced the rest of the genome with the inbred line genome and got to really see what is the effect of the Y chromosomes alone. And as we did this, we saw that females that come from these lines don't show anymore any body size variation, whereas the males do. So we have one group of males, so males that had that came from the large male selection line, their Y chromosomes basically cause them to have a so much larger body size that they nearly lose the difference to female size. So this showed us that we have kind of functionally two different Y chromosome haplotypes based on these phenotypic results. And then to try to narrow this down, what we did is that we did back biosequencing of these male Y lines. So we took these males here and we took these males here, and we sequenced their genomes, and we assembled the Y chromosomes. The way we did that is that in order to identify the sex chromosomes, because this had not been done in this system before, we leaned on the prediction here that the fixed differences that these haplotypes, these genomes should show, should be on the Y chromosome. In addition, we had Illumina data to compare the read coverage between the sexes, and then we also did PCR verification for the longest contig that we could find, and we could verify that it really was male linked. So through this work, we could see that the C. macrolactus Y chromosome actually has high gene density and a lot more genes than we would expect, for example, based on the knowledge from Drosophila melanogaster. So we find about 400 genes overall there. And what we find is that when we look at the fixed differences between these two different haplotypes, we find that there are about 136 genes that have fixed SNPs. But then we also find that there is a really strong candidate gene, target of rapamycin, that's been translocated from the autosomes to the Y chromosome. And we don't only find it in these lines, we also find them in 3 different populations that we've looked at since. And so to show you more, this target of rapamycin, it's a conserved growth regulator. It's found from bacteria to humans, and it's a strong gene affecting lifespan. It regulates cell size, cell number. It regulates apoptosis, and we find it that in females we find it only in one place in the genome. In males we find it in the. This has lost a lot of lines. This is supposed to show genomic links between the autosomal location in the 2 different haplotypes and how we find copy number variation in this Y-linked TOR gene, so that the males that have the haplotype that causes a small body size, they have 3 copies of this TOR on the Y chromosome, whereas the males with large haplotype have only a single copy. So this is a very strong candidate to explain this size difference, and it's really interesting. I haven't come across any studies where in any other system you could find this TOR gene in the sex chromosome. So just it being there is a good candidate to explain the sex difference, but this copy number difference is then that seems to be the strong candidate to explain this, and this is. Now our ongoing work is looking into the RNA-seq of these lines to see how this TOR pathway might be differentially expressed in these haplotypes. So then, if I have still some time, I'll talk a little bit about the sort of genomic signatures of sexual conflict, even if when the dimorphism evolves through this kind of major effect loci in the sex chromosomes, there is still a conflict present because of the vast number of other loci that affect body size variation and the shared genetic variance that we could see there. And so we're also interested in what signature these different forms of sex-specific selection leave to this shared genetic variance. And the prediction here is that sexually antagonistic variation is able to maintain genetic variance under balancing selection. And this is essentially a one-locus model. So we wanted to take a shot and see if our artificial sexually antagonistic selection would be able to also maintain the genetic variance in the body size genes. And what we did here is that we took the males in the sex-limited selection lines, where we selected also for smaller body size, because the male evolutionary trajectories here are very comparable. The phenotypic change is very similar in these lines, but the difference is in the response to selection in the females. Our first work here was repeating the pedigree analysis with new pedigree data collected after the selection was finished from these selection lines. And we could there compare the ancestral genetic variance to what then, how much genetic variance we could detect after finishing this sexually antagonistic and male-limited selection. And we could see that SA selection did maintain significantly more additive genetic variance as well as dominance variance. I didn't bring this up, but we also detected small but significant amount of dominance variance only in females. So both of these variances could be maintained better under SA selection. So this is really interesting because we're interested as evolutionary biologists about the mechanisms that can maintain genetic variance. And even if we didn't see any response to selection, SA selection in females, this genomic constraint that is present when selection operates antagonistically then is able to actually actively maintain genetic variance. So this puts SA selection up there as a one potential mechanism that maintains additive genetic variance in fitness-related traits. And we see a signature of this also in genome-wide. So we have pulse-seq data. I don't know what has happened. Something has happened to the colors here. So we have pulse-seq data from different time points in each of our selection lines, and we can see here that in this PCA analysis, the first axis of variance is really kind of recapitulating the selection history here. Here's the ancestor population. Sexually antagonistic selection lines are very much they haven't really diverged from the ancestor population, whereas the small male selection line is in one end, and then the large selection lines are on the other end. So this is ongoing work, but what we are now wanting to do is to really look for signatures of balancing selection and compare how the sites that are under directional selection here, how they might show signals of maintenance of nucleotide diversity under SA selection. So with that, I just want to summarize that the context in which body size evolves is as part of these life history syndromes. It's evolution, and overall these life history syndromes evolution is affected by this sexual conflict over mating between males and females. And we see overall that there is this faster evolution in life histories in males, and now it's really interesting with this Y chromosome here that to look into the phylogeny and see how much of this faster male evolution can be explained by the divergence in the Y chromosomes, and whether we find this TOR candidate gene explaining any of that variation in the sexual size dimorphism. So this really puts Y chromosomes on the map out there as a potential for actually maintaining additive genetic variance that is relevant for sex-specific evolution, despite of their small size. It's an ongoing work, and we're going to start to look into how exactly is this variation maintained. So hopefully in the future we can understand that better. And SA selection seems to be a potent force maintaining additive genetic variance in fitness-related traits. So thanks very much for your attention.